Hello everyone, my name's Dan Lloyd. I'm from the School of Biosciences at the University of Kent. One of my roles at the university is as the Programme Director for our MSc in Cancer Biology. So I suppose one of my key roles is developing the next generation of cancer researchers. I've been a cancer researcher for 20 years, and we need cancer researchers because cancer remains a very significant disease. It will affect 1 in 3 of us here in the UK, 1 in 3 in this audience. And so this remains a very significant health and social problem. So it's very important that we have cancer researchers. It's very important that we have people who have the skills and the expertise to understand what makes this cell here a cancer cell. And if we understand what makes cells cancer cells and how they differ from normal cells, then we have a much better chance of having the best possible treatments and therapies for those people that are affected by cancer in the future. So my interest in cancer started from research involved in understanding DNA damage. And damage to DNA is a very significant factor in cancer. We know that we are constantly, and in many cases unavoidably, exposed to DNA damaging agents within the environment. So I've got 2 examples here of many. Sunlight and cigarette smoke. Sunlight contains ultraviolet radiation. Ultraviolet radiation will cause chemical changes to DNA, and as a result of that, it is associated with a risk of skin cancer. Cigarette smoke and cigarette tar, in fact, contains chemicals that will damage DNA, and obviously because of the physiological nature of smoking, that has an enhanced risk factor for lung cancer. So DNA damage causes cancer. We know, however, from cancer chemotherapy that there are some very effective chemotherapeutic agents used to treat cancer. And those agents, for example, etoposide and bleomycin, these are agents that will damage DNA as well. So we can use DNA damage for therapeutic purposes as well. We can use it to treat cancer. Now the problem, as I'm sure many of you are aware, in terms of cancer chemotherapy, is that there are some really pretty debilitating side effects associated with some of them. And the reason for those side effects is that targeting toxicity and trying to kill cancer cells inevitably means that with cancer chemotherapy, you're killing some healthy cells as well. And for that reason, you will kill healthy cells that lead to some of the debilitating side effects like hair loss and loss of the lining of the gut, causing gastrointestinal problems. And so the challenge for us as cancer researchers is to try to understand a bit more about the cancer cell and the difference between the cancer cell and a normal cell, so that we can perhaps target that toxicity a little bit more effectively, target cell death towards cancer cells while leaving healthy cells intact. And one of the things that's emerged over 20 or 30 years of cancer research has been some of the things that make cancer cells different to normal cells. And one of those things is that cancer cells tend to express very high levels of certain proteins on the surface of the cell. And they tend to be in much higher levels than we find in normal cells. So this is what cancer researchers revealed to us over the last 30 years or so. And I've got some examples of those proteins here. I'm not going to run through them. This is not an exhaustive list. There are many, many proteins that have been identified. But one of the things that you'll see is that some of these proteins are associated with particular cancers: colorectal cancer, lung cancer, breast cancer, etc. So cancer is a family of diseases, not just one. And some of these cell surface proteins are characteristic of individual cancers. I'm going to focus a little bit on one of these, this protein here called CD33. Now this is a protein that's found on the surface of acute myeloid leukemia cells. And acute myeloid leukemia, or AML, is a particular kind of leukemia affecting a particular type of blood cell. And so theoretically, we would be able to target this CD33 protein and therefore be able to have a targeted therapy towards acute myeloid leukemia, leaving healthy cells intact. That's the concept anyway. The question then is how you target that protein. How do you target a protein that's specific to cancer? And there are a number of ways of doing this, but I think the mechanism that I'm particularly excited about is using antibodies. Antibodies are a feature of our cells anyway. We use them in our immune system, and one of their key roles is to use this. This is an antibody. It's a Y-shaped molecule, as you see here. And in the top left and right, we see the region here in red that is actually responsible for binding to proteins that are found on pathogens such as bacteria. And an antibody will work by using this region to bind to the protein on this pathogen, and then the rest of this antibody, which we're not going to talk about much more, is responsible for integrity of structure, but also importantly recruiting other elements of the immune system, such as white blood cells, to the complex that's been formed so that that pathogen is ultimately destroyed. Now, one of the advances in molecular biology in the last few years has been to alter antibodies so that we can actually change these sequences. And these sequences are variable because that allows antibodies to have the capacity to bind to different kinds of protein. But actually we can engineer that now. We can develop proteins, engineer them so that we can engineer antibodies so that they will bind to proteins of interest, and that might include proteins that we find on the surface of cancer cells like CD33. So here's what we would hope to do then. This is a very simple diagram to illustrate just a cancer cell and a non-target normal cell. You'll see the cancer cell, for example, here has CD33 on the cell surface. And the idea would be that the antibody comes along and binds to CD33. It's therefore bound exclusively to the cancer cell, because you'll see here the non-target cell doesn't have CD33 and so is unable to bind. And increasingly, just from the antibody perspective, once we've engineered these antibodies to bind to specific proteins, we're able to use them for diagnosis. And increasingly, in biomedical laboratories in hospitals, they will use antibodies to be able to diagnose specific types of cancer. We don't stop there though, because if we've got this targeting mechanism, why not use that and try to develop a therapy? And one way that you could do that is by attaching something nasty to this antibody, using the antibody as essentially a delivery mechanism to this toxic agent that would hopefully kill the cancer cell. So we start again. Here's our cancer cell, CD33 in that red triangle. In this case, we've bound an antibody, but the antibody is attached to a toxin. And the toxin, because it's been delivered close to the cancer cell, is able to exert its toxicity on that cancer cell. Because the non-target cell doesn't have CD33, the antibody is unable to bind. This toxin is not in sufficient proximity to the cell to be able to have any influence at all. So the only cell that dies on this slide is the cancer cell, because it's the only one that's been affected by the toxin. And that's exclusively about the delivery of the antibody to that CD33, the tumour-specific marker. So we can use toxins. I'm particularly excited, though, about the use of radioactive isotopes. And the reason is, as you'll see in a moment, there's a great deal of versatility in terms of the types of radioactive isotopes that you might be able to use, depending on the different type of cancer that we're looking at. But also, the way in which radioactive isotopes kill cancer cells is by damaging their DNA. So it goes back to my original expertise and interest in DNA damage and its association with cancer. But we can hopefully make it a little bit more targeted here, because once again, here we have an antibody. It's bound to CD33. The antibody is attached to a radioactive isotope. That radioactive isotope emits radioactive emissions. These tend to be particles, and these radioactive emissions will target the DNA and cause strand breaks, and that causes toxicity to this cancer cell. And once again, because the non-target cell is non-bound by the antibody. It doesn't have CD33. The antibody can't bind. This radioactive isotope is not close enough. It's not close enough to the non-target cell to have any influence at all. So once again, we've selectively killed the cancer cell and hopefully left the non-target cell intact. So that's the concept. Of course, I'm showing you a very simple representation of non-cancer cell and cancer cell there. But in the tumour environment, things are rather different. It's much more likely that you've got a complex model here, a mixture of collection of the blue circled cells here, which are the tumour cells, and the non-target healthy cells in pink. And we have the blue cells here in the centre. Now, we have versatility in terms of our choice of radioactive isotopes that you might conjugate your antibody to. And if you're treating a solid tumour, you need something that's going to be able to deal with that mass that we see here. And for that reason, we use beta emitters. These tend to travel, the emissions from beta emitter isotopes tend to travel over a range of millimeters. And that's quite nice for a solid tumour because it's going to get far enough into the tumour to be able to have some kind of difference. But also, as you see here, if you bind enough of the antibody and then you get the radioactive emissions, you see here that you kind of get this crossfire effect that concentrates all of that radioactive energy into the centre of the tumour. And sure, it may be the case that an emission might go in the opposite direction and have some kind of impact on healthy tissue, but it's only going to travel a few millimetres, and it's not going to have the same kind of concentration as you see in the core of the tumour because you don't have the cell-specific marker that's allowing the antibody to bind. So this ought to have a much better therapeutic outcome than cancer chemotherapies, and certainly there are antibody beta emitter radioconjugates being used in the clinic currently. Now, not all cancers are solid tumours. If you think about a leukemia is actually disseminated through the body because it's in the bloodstream, it's in the bone marrow, and the tumour cells are disseminated all among healthy cells. So this model's not going to work here. We have a much different tumour model. We have one that's much more like this, so that your blue cells and pink cells are all mixed together. And in that case, the crossfire effect that we see with beta emitters is not going to be any use at all because caught in the crossfire are going to be those healthy cells. So in this instance, we need a much shorter path length radioactive emission. And so much more advantageous here is alpha emitters. They don't travel very far. They only travel in the micron range, maybe 1 or 2, 3, maybe cellular diameters. And what that means is that we can target, if we do target the antibody to cancer cells via CD33, for instance, then the emission is not going to go very far. It's not going to affect any other cells apart from the one that has been affected. So here we have the antibody bound to a tumour cell. It's deposited all of its energy within that cell. It's damaged the DNA and hopefully kills that cell. But because of this short path length, none of the pink cells are being affected here. So once again, we've got a very selective targeting, but only because we've got the right coupling of antibody and the appropriate radioactive isotope with the appropriate path length. So, as I mentioned to you, we have antibodies that are able to achieve this, but I said to you, the important part in terms of what we've been talking about here is this variable region at the top that's responsible for binding to those proteins, those antigens that are involved in cancer. And one of the things that we've learned from our advances in molecular biology is that we can slice and dice this, this antibody and engineer it so that actually all you have is this top portion. You don't need the rest of it because we're not engaging with any of the rest of the immune system that our body has to. And so if you add a linker sequence here, a sequence of protein that links those two regions together, you can actually produce this as a single gene in very simple model systems like bacteria and yeast, as opposed to much more complex systems that are required for intact antibodies. And that has a number of advantages in that, firstly, it's cheaper. Antibodies are very expensive to produce. It's cheaper, it's quicker, we can produce high yields, and potentially the smaller size has some advantages in terms of its delivery to different parts of the body, such as tumours. So actually myself and collaborators have we've engineered a smaller fragment that we saw there. It's called a single chain antibody, or single chain FV, which is just that top red portion of the antibody. And we've produced it. Here it is. So here it is on a protein gel. We're able to isolate this protein and analyze it. And one of the things that we need to do is verify that this smaller fragment is still able to bind to CD33 present on cells. And I have that analysis on the next slide. This is some microscopy that looks at different elements of the cell. It's looking at the cell membrane, first of all, labelled in green. It's looking at a fluorescently labelled version of our antibody, and that's shown here in red. And then it's overlaid those two images on a microscopy technique that we use in the School of Biosciences called confocal microscopy. And what you can see, hopefully, is that not only has this anti-CD33 single-chain antibody bound to these CD33 positive cells, it's actually gained access to the cell. It's gone inside the cell. And this is not the only analysis that we've done here. The confocal microscopy happens to generate quite beautiful images. We've verified this using other mechanisms, but the fact that this is going inside the cell actually offers a great deal of versatility, because it might mean that we can use some different types of isotopes that will take advantage of that real proximity to the DNA contained within the cell. And it potentially allows us to use some toxins that are only active if they're present within the cell. So I'm reminded here, this kind of access that this antibody has gained to the cell, I'm reminded of when I was at school. When I was at school, I was told the story of the Trojan Horse. It's a bit like our antibodies. I kind of think our antibodies are behaving a bit like the Trojan Horse, bringing in this toxic cargo in close proximity to cancer cells, and potentially also bringing it inside the cell, which is really an interesting way of thinking about how we treat this sort of subterfuge way of treating this very sneaky disease that tends to find different ways of evading existing therapeutics. So hopefully I've given you some insight there in terms of how our knowledge of cancer cells and what makes a cancer cell different to a normal cell can really inform us think carefully about how we can use the biological sciences to develop new, better therapies for treating people with cancer. Thank you very much for listening.